Cycas, male and female gametophyte. In our previous lecture, we studied about reproduction in the sporophytic phase, and moving on forward, we are going to study about the gametophytic phase in Cycas. Cycas is a heterosporous plant. It produces two types of spores, microspore and megaspore. Microspore develops into male gametophyte and megaspore develops into the female gametophyte. The spore represents the last phase of the sporophytic generation and it gives rise to a new gametophytic generation. Microspores are the male gametophyte. Microspore germinate within the microsporangium before dissemination. Development of microspore occur in three phases. Development of male gametophyte before pollination, which occurs partially inside the microsporangium at the time of pollination, and development of male gametophyte after pollination in the pollen chamber of the ovule take place. Therefore, there are three phases in which the microspore or male gametophyte develops. Moving on to the development, as we can see that microspore is a round structure with an internal wall called intine and external wall called exine. It undergoes a transverse division where it forms a small prothelial cell and a large enthedial cell. Further, enthedial cell undergoes a transverse division and forms a small generative cell and a tube cell. Here, the microspore becomes three-celled and it is dispersed. So, the microspores are dispersed at the three-cell stage and are pollinated. This is the completion of the first phase which occurs inside the microsporangium. Now, after dispersal, we come to pollination, where the microspores are shed in the air after dehiscence. At the time of dispersal, some of the cells of the nucellar beak present in the ovule are disorganized and form a viscous fluid, which oozes out of the micropyle and forms the pollination drop. Thus, pollination drop functions to stick or entangle the microspore. After some time, pollination drop dries up and the microspores, after they are sucked into the pollen chamber through the microbial canal, the viscous fluid dries up, plugging the ovule and not allowing further entry of any further pollen grain or microspore. After the pollination, the ovule increases in size and the unpollinated ovules get dry up. Now, third phase is the development of male gametophyte after pollination, where it enters the ovule in the pollen chamber. As we discussed that after three-cell stage, the generative cell divides into a body cell and a stalk cell. And the wall we discussed, the exine ruptures and the intine protrudes out in the form of a pollen tube. The pollen tube penetrates the nucellar tissue and grows towards the female gametophyte. The stalk cell does not divide, but body cell divide to form two male gametes and form entherozoites. Just before fertilization, the entherozoites swim freely in the cytoplasm of the pollen tube, and there is an interval of about 4 months between pollination and fertilization. Finally, the male gametophyte forms the male gametes called entherozoites, which are naked. The blepharoblast of the entherozoites elongates into a largely spirally arranged structure with many cilia. Thus, as we study the development of male gametophyte, which forms male gametes, And finally, these male gametes will fertilize the egg. Moving on forward to female gametophyte. So, female gametophyte develops from the functional megaspore, which is deeply situated inside the ovule or megasporangium. The nucleus of the megaspore divides by free nuclear divisions to form a large number of nuclei, and simultaneously it also increases in size. A vacuole develops in the center of the megaspore, which forces the cytoplasm towards the periphery, and the vacuole contains a whitish fluid which is formed by disintegration of the cells in the vacuole region. Moving on forward to understand the development of nucellus, slowly the formation of cell wall starts from the periphery and extends toward the center. The tissue thus formed is called female gametophyte or prothallus. Finally, the cells of the endosperm are haploid, and the nucellus is used by the gametophyte and is presented as a thin layer in the mature ovule. So, we discussed that the functional megaspore forms a female gametophyte where the megaspore increases in size and undergoes a free nuclear divisions, forming many nuclei. These nuclei are shifted toward the outer side at the periphery, forming a vacuole at the central region. Slowly, the wall formation starts from periphery to the center, forming a endosperm tissue, which is called prothallus and female gametophyte, and it is haploid in nature. Now, development of archegonia, which is a female reproductive organ which develops from the superficial cell of female gametophyte at the micropylar end, and these cells function as archegonian initial. These archegonian initial divide by a periclonal division to form an outer primary neck cell, and which divides by vertical wall to give rise to two neck cells, and inner central cell, which enlarges in size and its nucleus divides to form a small ventral canal nucleus and a large nucleus. Therefore, this archegonial initial undergoes a pericranial division to form a primary neck cell and a central cell. Primary neck cell undergoes a vertical wall division or anticranial division to give rise to two neck cells. Inner central cell enlarges and divides to form a small ventral canal nucleus and a large egg cell nucleus. The egg of the cycas is largest among the living plant. The mature archegonium consists of 2 to 4 neck cell and egg. The number of archegonia in ovule may vary in different species of cycas. We can see here the primary neck cell divides by anticlinal division to form two neck cells. Here these are the jacket cells and a central cell which divides into a egg cell nucleus and a ventral canal nucleus. After the formation of egg cell and the male gametes entherozoids, fertilization occurs where pollen tube after reaching the archegonial chamber breaks the nucellar passage and pollen tube bursts and releases its content in the archegonial chamber. When male gamete comes in contact with the neck cells, it gets sucked violently and the male gamete enters the archegonia where the nucleus fuses with the egg and the zygote is formed. The phenomenon of entrance of more than one male gamete in the archegonium is called polyspermy, which is noticed in gymnosperm and is a frequent phenomenon in gymnosperm. After the formation of zygote, formation of embryo occurs where the diploid zygote functions as the mother cell of the sporophytic phase. because after its development, it will form the sporophyte of the Cycas. It increases in size, undergoes free nuclear division, as a result of which free nuclei are formed. A central vacuole appears and the nuclei are shifted to periphery. The cell wall formation starts, which extends from the basal region. It initiates in the basal region and extends to the upper region. This cellular structure is called as proembryo, which is differentiated into three regions: the uppermost postural region, middle suspensor region, and the basal embryonal region. The posterior region absorbs the nutrient from the micropylor end and provides them to the developing embryo. The suspensor region elongates and becomes coiled, which puts the embryo deep into the female gametophyte, and the embryonal region forms the main part of the embryo, takes about 1 year for complete development. After some time, the two cotyledons are differentiated and the plumule is formed in the central depression between the cotyledons, forming a well-developed sporophyte. So, the embryo enlarges, ovule increases in size, the mature seed is red, orange, or dark brown in color, surrounded by three layers of integument. The outermost layer of integument is fleshy and is called sarcotesta. The middle layer becomes stony and hard, is called sclerotesta. The inner layer remains thin and papery, which forms a complete seed structure. The germination is hypogeal and the cotyledons remain inside the seed and the soil. The seed absorbs the water through micropyle. The radicals form the primary root and a plumule develops into a leaf-bearing shoot, forming the first crown of foliage leaves after several years and therefore developing into the well-developed sporophytic phase. Talking about life cycle of Cycas, starting with sporophytic generation, where we discussed that the oospore or zygote is the mother cell of this sporophytic phase. So, a zygote develops into an embryo, forming a seed and seed giving rise to a well-developed male gametophyte. As we know, Cycas is a dioecious plant, a gymnosperm. So, we get a male Cycas and a female Cycas, which forms a male cone and a female strobilus respectively. As cones are not formed in the Cycas, female cones are not formed in Cycas, so we represent them as megasporophyll only. Male cones form microsporangium and female strobilus form megasporangium. Megasporangium has a megaspore mother cell which divide by meiosis to form megaspore, and microsporangium has microspore mother cell which divide by meiosis to form microspore. Microspores are a male gametophyte which give rise to male gametes called antherozoids, and megaspore forms the female gametophyte called embryosac, which has a female gametophyte prothallus called as prothallus, which forms a female gamete called egg. These antherozoids and egg nuclei fuse with each other to form or fertilize with each other to form a oospore or zygote, finally forming an sporophytic phase again. So this is the life cycle of the cycas. Here we can see a pictorial representation of the life cycle of cycas, where the megasporophylls and the microsporophyll have microsporangia and megasporangia respectively, forming megaspore mother cell, microspore mother cell, dividing by meiosis to form microspore and megaspore, which forms male gametophyte and female gametophyte respectively, forming male and female gamete, which fertilize or fuse with each other to form a zygote. Thank you.